So our next speaker is Eric Morgan. He is the Chief Operating Officer and co-founder of Bioage Labs, which uses AI-based drug discovery and systems biology to target the pathways that affect aging. Bioage's drug discovery platform is longitudinal in nature, and it consists of the personal medical records and eventual longevity outcomes of a large number of human beings in order to create a detailed map of how we age, including proteins, metabolic issues, and genetic transcription. If you'd help me welcome Eric Morgan.
All right, thanks everyone. It's a pleasure to be here. So I'm going to talk today a bit about our platform, how we find targets, how we validate those targets, and then an example of taking one of those through animal validation and then also clinical trials. I'll share a little bit of data that you may have seen in a press release recently. So at Bioage, we're a platform-driven clinical stage biotech company. We're building a portfolio of therapies that target fundamental mechanisms of aging, but we're employing those first to treat severe diseases. We have a human-first discovery platform. That means we use human data to find the targets that we're going to try to modulate in humans. And we have a proven track record of results. We'd like to change the nature of aging. We'd like you to consider this idea. What could you do with more healthy time? I think we're all thinking about that at this conference. Aging's important. I'll leave it at that. It drives a lot of diseases. Clearly, there's not a very low upper bound on longevity, so let's reach for more. And this is sort of profiling the growth and interest in aging in recent decades. And just highlighting mostly that it's only been in recent years, over on the right where the human figure is, that we could really study aging productively in humans, because aging in humans happens on the scale of decades, and it's observational, not experimental. So it's really only by collecting data from people over decades and being able to interrogate it in a high throughput fashion using modern omics methods, and then analyzing that data using modern analytic methods, that we can really start to learn a lot about human aging, and that's what we do at Bioage. So a very useful way to think about what we do is that when you look at the population around us and the population in these healthy aging cohorts, there's a wide range of outcomes. Some people are living a really long time and they're really healthy until they die, and other people have a much worse trajectory. And it's sort of investigating these natural examples. What's the biology that separates these people, sort of when they're all in healthy middle age, they have these future trajectories that are based on how their biology is operating. And so we recruit these healthy aging cohorts, interrogate them, and ask that question using the data. What's different about the biology of people who age really well, and how can we modulate that to improve human health? There's a lot of data coming out of these cohorts, and it's a data-driven process. It's a hypothesis-free process. So we get a really interesting look at the biology of aging over decades in people. What's really important when you're doing this kind of cohort-based work is what outcomes you focus on. How are you measuring aging? And so we're looking at things like all-cause mortality and longevity, but also really critical measures of health span, like your cognitive function, your physical function, when you get different diseases. And so these are the kind of rich phenotypes that we like to have in our cohorts to help us make sense of the biology. And here's just an illustration of kind of the first level when you take a whole bunch of individual molecules, in this case 5,000 proteins that we're seeing in the middle there, each represented by a circle. And each one of those proteins has its own associations with different phenotypes. For example, on the left, grip strength, cognitive decline, renal function, cardiovascular function. Each of them has an association with mortality. So this just starts to give you a sense of how many different associations we're working with, even on an individual protein level. And then, of course, we can learn about the interactions, the relationships between these different proteins. So in healthy aging cohorts, where we see this biology changing as these people age, what can those tell us, again, in a hypothesis-free way about how is the biology working. So we map those relationships, we can sort of cluster that data, we can allow the data to tell us what are the interesting clusters that exist there, and then interrogate those clusters afterwards. And the interesting thing about this, of course, is that we find all sorts of things. Not only do we see what we might expect to see, things we can map directly to known mechanisms of aging, which are really cool because we have a better chance of understanding what's going on. But we can also actually see past this circle into I guess what we've playfully termed the untracked wilds over there, where it's just signals coming out of the data, and we don't know how they connect to known aging biology. But hopefully we'll find out. So at Bioage, we have 3 focuses in terms of clinical development. Where are we taking these therapeutics? Immune aging, muscle aging, and brain aging. These all have high unmet need, and they allow for the right kind of clinical development, where we can start with initial high impact, short clinical trials, and we can go on to really big parts of aging that affect all of us. So I'm going to give you an example. The Apelin example, the 105 case study, how this came out of the platform, what we did to validate it, and how we got a molecule, and the clinical trial that we ran, and then sort of what our future plans are. So this is a molecule where we were very interested in finding something, a therapeutic that would work for muscle aging. So we created a score that integrates different aspects of both longevity and muscle and physical function, and we found a protein that was on the tail of that distribution and also had an interesting molecule available commercially that Amgen had that they discontinued that was nice and safe. And so we looked harder at that. We're like, this is an opportunity, potential opportunity here. And we found on the lower right there, you can see that there's a really interesting relationship between circulating levels of apelin and both preservation of grip strength and longevity, the chance of living past 85. So that was really interesting. So we went ahead and did some animal experiments. And at the bottom here, the first thing we did is actually look at molecular level relationships. So what's happening at the pathway level. So we actually went into that map that I showed you earlier that we draw just based on the observed interactions. And we found the sort of apelin superpathway and confirmed that at a pathway level it has perhaps even a stronger relationship with mortality. But then we went on to animal validation. So we have observational human data telling us something really interesting, that maybe this is an actionable human pathway we can modulate to affect muscle health in people. But let's do an experiment. So we took old mice and we casted their legs, and you can see you cast one leg, you don't cast the other leg, and of course the casted leg can't move, it gets substantial atrophy. And what you can see on the graph on the right there is that if you get the drug in purple, the mice don't lose any muscle at all. So that was a really interesting, very unexpected result in its magnitude. And so we went ahead and based on that sort of accumulated data and some other animal experience, we decided to actually in-license this drug from Amgen. We call it BG105, and just for reference, we have both an oral formulation and an IV formulation, so it can be a pill or it can be an injection, and it came in with a clean safety profile from phase 1. And so we designed a study, actually just a little more background on apelin. So it's really interesting in that one thing that has become known about apelin is that it's produced by muscle and other tissues. It's also perceived by muscle and other tissues. And one thing that we know it does is that in the presence of apelin, you have improved muscle repair, increased muscle mass and strength in animal models, and increased energy production. And it's clear that as animals and people age, this activation goes down, and so it's a very simple sort of hypothesis here. Let's fix that by activating the pathway in older animals and people. And we collaborated on a number of animal experiments with an Apelin expert, Dr. Dre in Toulouse. And so he helped us establish that we're mobilizing stem cells and improving muscle repair in response to a cardiotoxin injury. So really interesting evidence that we're able to improve stem cell mobilization and regeneration. Oh, here's the details of the experiment I was talking about on the right. Additional experimental details. So on the right is a grid hang test after animals had been treated for 60 days, which is the data shown on the right, with this drug. So we saw that just on the left is wheel running. So if you just take naturally aging mice, so you take 20-month-old mice and you give them this drug over 60 days, what's really interesting is that they preserve their ability to run on the wheel by quite a large magnitude at the end of that study after only 2 months of treatment. And at the end of that, we had asked them, put them on a grid and saw how long they could hold on, and we also saw a significant result there. So a number of really interesting results. So it was this combination of results, and here's that atrophy data again, that convinced us to in-license this drug from Amgen and design a clinical trial that really parallels that casting trial, where we, made the mouse limb immobile, saw substantial atrophy. In this healthy older volunteer study, we put volunteers, subjects at bed rest, and this is a well-established intervention. It's been studied in the literature. They lose a lot of muscle. The older you get, the more muscle you lose in this period. Patients were either given the drug or they were given a placebo, and we observed them over 10 days, and we measured sort of a number of metrics of muscle size and function, as well as safety and PK and biomarkers. And what we saw was really interesting, was that basically the patients on placebo had the loss of muscle that you'd expect, and the patients on drug were protected from this to a great degree. And so you can see these percentages on the bottom there. So almost no matter what sort of metric you use, whether on the left you take the circumference of the limb, just using a tape measure, this is the thigh, or use ultrasound to measure the individual muscles within the thigh. We focused on the thigh because it's a weight-bearing limb that's known to have a lot of muscle loss in bed rest, or whether you actually use that ultrasound to look at the muscle itself, like the texture of the muscle, the quality of the muscle. You can use ultrasound to essentially assess fat infiltration and replacement in muscle, which happens during atrophy. And so we not only showed that we prevented the muscle loss, so for example, the circumference where we measured the leg with a tape measure, all of the loss was prevented. That's what 100% means there. And the p-value was less than 0.001. When you measure the actual vastus lateralis muscle, it was a 73% protection from that decline. The cross-sectional area was a 58%. So now we're bound to the muscle quality. So in the treated group, we had only 1 out of 11 patient decline in muscle quality from the best rating to the one right below that, so a little bit of fat infiltration, whereas in the untreated group, 8 of 10 patients declined in, again, not only their muscle size, but their muscle quality. So, and then on the right, we also took biopsies of the muscle and used a fairly innovative method developed by Bill Evans and Mark Hillerstein out of University of Berkeley, where you give patients deuterated water and use that to essentially label the creation of new protein, and you can measure muscle turnover. So this is a proteomic method to figure out how much muscle you're synthesizing when you take a muscle biopsy. And when you go on bed rest, you have a massive drop in how much protein you synthesize, and we showed that in this study. And when you're on the drug, you have much less. You're largely protected from that drop again, and it sort of provides one mechanistic clue as to how this is happening, how we're being able to preserve muscle mass in these people. So this was a really exciting result. And we're actually going to be hosting a virtual event on January 4th, which we invite you all to register for and attend. Our press release, you can easily find, has the link. Our CEO is talking along with Bill Evans, who is a consultant for Bioage and just a world expert on sarcopenia and muscle aging. Paul Rubin, our chief medical officer, and Ewan Gallagher, who's a professor in the University of Toronto and an expert on, among other things, diaphragm atrophy. So where we plan to go with this drug is we've shown you can protect patients from muscle atrophy. And so there's a place where patients experience really severe muscle atrophy and it's really awful for them, and that's the ICU. And so when patients go on a ventilator, on the left here, their diaphragm atrophies very quickly, within days. And that atrophy is associated with really high risk of not being able to ever get off the ventilator, as well as poor clinical outcomes generally. And at the same time, whether they're on a ventilator or not, they're in bed and they're really sick and they lose a lot of muscle. And that's a really big problem that was highlighted recently amidst the COVID pandemic, right? That people get really massive atrophy and they can have really reduced function and never get back to their baseline. So we're going to test whether we can prevent both of these things, whether we can help get people off the ventilators and maybe whether we can help them not have this awful muscle atrophy. And just, a couple of key stats on the left, you can see that if you have diaphragm atrophy on the bottom, you have a 4 times higher risk of in-hospital mortality, 27 versus 7%, so that's bad. And on the right, if you have critical illness myopathy, which is this peripheral wasting, due to being in the ICU, you have again like a 20% increase in mortality in hospital and at 5 years. So really bad, both of them. So we'd love to find a solution to this. And these are steps on the way to where we think this drug has really amazing potential, which of course is in age-related muscle loss and loss of function, which is sarcopenia and frailty. So we're really excited to move towards that path. Yeah, thanks for listening.
I have a quick question. This is really, really exciting, obviously. So big congrats.
We're very excited. Thank you.
Yeah, big congrats on that. Awesome. So my question is on how you decided to in-license the drug as opposed to modify it, make a different version for yourself, or like we heard from Hannity earlier, like have it just be a peptide as opposed to a small molecule.
Mm-hmm. Mm-hmm. So peptides, that's a strategic decision. Peptides are, in this case, would have been IV only and have a very short half-life. So there are some challenges there, which makes sense in depending on the clinical scenario, but not for us and not for where we're going at the moment. And in licensing generally, it allows you to move a lot faster if there exists a drug, small molecule, biologic, whatever, that already hits the target you're interested in and already has human safety. That's just an awesome scenario to be in if you can get a hold of that drug. So I think probably most people would like to jump ahead if you can, and it's really just a matter of being in the right circumstance where you have that option and that drug's available and all those things. So it's usually good. You're kind of stuck with whatever already exists because a lot of work has gone into that and taking it through the regulatory path, so you can't change it. So you can make your own from scratch, which we're doing some of that too. It just depends on the circumstance. They're both good strategies. Mm-hmm. Sure, I think we had one back there and then here. Yeah. Mm-hmm.
Could you comment on the clinical trials from Amgen and the indications and why do they fail?
Right. So Amgen was looking at this drug previously for heart failure. So Apelin also is perceived by the heart. They didn't tell us exactly what they thought of it, but looking at it, it looks like they just decided it didn't have enough of an effect in their indication to want to pursue it. But I can't comment on their actual decision process. So we decided to take it into muscle because of exactly the animal evidence that I was showing you. There were also some papers exploring apelin and muscle function, so that kind of pointed us to those experiments. And we did a number of experiments, and the one that had the most interesting kind of potent result was that atrophy experiment, and that's why we went straight from that to the equivalent human clinical trials. We're proving the mechanism. We can prevent atrophy. We can do it really well, surprisingly well. And so that's how we got where we are.
Do you think there's any chance, I mean, heart is a muscle, right? So do you think there's any chance that a long-term treatment would have a positive impact on the heart as well? I mean, I'm assuming that they've done some kind of post-ischemia test, but see where I'm going?
Yeah, I don't think there's a reason that this might not have an effect long-term in the heart, for sure. Yeah, go ahead, yeah.
Yeah, hi. Really amazing data. Looks great. Just out of curiosity, what was the actual effect size in the thigh circumference between the two groups? Is it that, if I understand correctly, your treatment, they had no loss.
That's right.
But then what was the actual loss after 10 days of bed rest?
Right. So we're going to go into the full data, obviously, in this event. So if you want to go very deep, you can come along to that event. I'll say that these kinds of trials generally, where people do bed rest over 10 days, it's not unusual to see more than 5 to 10% loss in various measures of the lower limb, and our data was kind of consistent with that. So they lose a lot of muscle in a short time, and at least with the thigh, we were able to prevent all of it with that thigh measurement. Yeah.
Hey, fantastic job, and I'm really excited to hopefully learn more about this, and just really excited to see, success, right, with your company and with the first kind of clinical readouts. My main question is just on, were you guys able to measure any sort of functional correlation of the preservation with muscle mass? Is that going to be sort of required, showing improvements in certain strength metrics to get approval? And then the second question is, how are you guys kind of navigating the scenario with sort of hospital, right? Because would this be a treatment that patients would be taken, would only take if they're actually in the hospital? And, how does that affect sort of like the insurance reimbursements?
Very practical questions,
Yeah.
Kennedy. So far it's just a phase 1A. So far we're just proving mechanism, and so we haven't done much investigation of function because we were sort of had to pick and choose what we were going to do in this trial. Ultimately, I think the FDA has been telling us that if you want to get a drug approved for muscle function, that you do need to show not only a change in, actually, you don't really care about whether you change muscle size, I think. I think we care about that because that's on the path, but also strength and function. So I think we are going to have to show that ultimately in whatever indication this ultimately goes forward in, yeah. And so for the ICU, it would be in hospital. I think, there's a world in which it's only in hospital, and there's a world in which it starts in hospital and continues once you're home. Certainly for, I think, the peripheral, wasting, right now it takes them months to years to recover, so I wouldn't be surprised if we had to treat for longer. It could be both. And then, and reimbursement. There's a lot of implications. We can talk separately. Go ahead. Yeah.
Hi, I will echo everybody's congratulations. This is amazing for Bioage and for the entire aging community. I have a question. It's you initially said that this drug can be also administered orally. Why did you design this clinical trial with actually the IV formulation? That, of course, it's less patient-friendly.
So this is kind of this is a phase 1 inpatient trial. So that's when you have the most flexibility about what kind of method, how to administer your drug, and when you're trying to reduce the variables as much as possible, which is what you can do in an inpatient study. One thing that's really nice about the IV formulation is that the patient gets exactly what you give. There's no intervening process. Oh, some didn't, he lost one pill, other pill he ate and didn't get fully digested, didn't get fully absorbed. So it's a lot more consistency in terms of, how much drug's on board for you to be able to, figure out how much of an effect you have. Yeah. But ultimately, in hospital, it's nice ultimately, like these other indications, to have an IV formulation, because in the ICU, patients often aren't taking anything by mouth. But yeah, if you're at home, you don't want IV, you want oral. So it's good to have the flexibility for us. Yeah, cool.
Okay, this is the last one.
Last one here, okay.
Hi, thank you. Great talk. I just wanted to ask you if you guys have any data already preclinical that might show that not only that prevents muscle wasting, but that also could like help recovery.
Yeah.
So once a patient, does lose muscle mass and say that they go to PT, right? Could you use your drug to like shorten the PT, for example, training?
Yeah, so one thing I'll point to is I already talked about data that shows that if you injure the muscle with cardiotoxin, we have increased regeneration as well as mobilization of stem cell factors. So I think that is suggestive. And so far we've been kind of lasering in on exactly what we have the most evidence for. But I think that's an area we'd love to explore more in the future, because both are really useful in different contexts. Absolutely. Thanks. All right, thanks everyone.